So let's see. By the way, the recording is always here, so if it's ever not on, please remind me. So, speaking of oncogene and tumor suppressor gene, before we even speak about these things, we need to understand what is happening, what is the process that is happening. I sent a few photos before the class just to get an overview about what's up, what is happening. So there is this thing happening. You have in your body tumor suppressor genes and oncogenes. Oncogenes want to kill you, want to cause cancer. Tumor suppressor genes are preventing the oncogenes from causing the cancer. And the tumor suppressor genes are always fighting with the oncogenes. If you will have more oncogenes than tumor suppressor genes, you will have something called genomic instability, which will lead to cancer. If you have less tumor suppressor gene than the oncogene, that also leads to genomic instability, which also leads to cancer. So cancer can be due to 1. Overexpression of oncogene. 2. Destruction of the tumor suppressor genes. So what are oncogenes? There are so many different types, and sadly you have to know all of them. Not all of them, most of them. And it goes something like this. If you look at first aid, they give you in this fashion, they give them for you. I have to say, I don't prefer this fashion, but I like this page from first aid. So what I'm going to advise you to do, you must understand how each process happened, but overall print this page and stick it in a place that you are going to observe constantly, like every single day. For example, put it in your bathroom or in your living room. I don't know, some place that you see multiple times a day. Since it's really difficult to memorize every single portion of it. So, the tumor oncogenes. So this tumor oncogenes basically gets mutated. Originally, they are pro-oncogenes. They are not an actual cancerous yet. And I'm going to send a picture just in a sec. You can see on Telegram. So as you can, as you can see, there is this cells. It's a normal cell. So we have it. So this cell does something like this. The first thing to happen is some agent comes and affects the cell to lead to the cancer. For example, it can be a virus. Can virus cause cancer? Yes. What else it can be? It can be one of the oncogenes we spoke about yesterday. Any of these, ionizing radiation, that is correct. Anything can lead to mutation of the DNA. That can lead to cancer. Well, is it that simple? Well, yeah. It's more complicated, but you get the idea. It activates the oncogene there, and that basically you initially had a pro-oncogene, which was in your genome. This proto-oncogene, remove the proto from it. Why? Because of the risk factor you got, because of that trigger. So the trigger remove the proto-oncogene and convert it to an activated oncogene, which lead to cancer. This is in a really simple way to say it. Now we add some stuff. Speaking about these guys, an oncogene can be in the following ways. It can be a problem with the receptor. It can be a problem with the cell cycles regulators. It can be in many things. But that's like the other one is actually tumor suppressor gene. Just a second, someone is joining. Okay, so speaking about this guy, let's tell you what is happening actually. If we imagine that you have a cell. This cell on its wall, it has a receptor. For what? Well, I don't know, multiple things. It can be growth factor, for example, okay? Or it can be a receptor tyrosine kinase, for example. And then inside of it, it have what? A secondary messenger. And then it has a genome. And the genome get expressed. And after it get expressed, which include transcription and the translation, it gets released. This secondary messenger, there is many of them. One of them is called the RAS system, the RAS GT base. And today we are going to speak about all of them. Speaking about oncogene, it's a gain of function mutation. Why? I told you the proto-oncogene has become the oncogene. So it's a gain of function mutation. You initially had the proto-oncogene, and now it has become an actual oncogene, which increases your risk of cancer. So you require a damage of one allele of a proto-oncogene. What do I mean by one allele? Well, your chromosome is normally consistent of two portions, like two chromosomes. Yeah? So for each gene you have here, you will have an analog gene for it on the other chromosome. To get cancer, in case of the proto-oncogene, you only require one allele of these two alleles to have the basically the mutation. In contrast with the tumor suppressor gene, in which you have the two, you must have the two alleles to be destroyed to get the mutation. Okay, speaking about the oncogenes, it requires damage to only one allele of proto-oncogenes. There is many types of them. As I told you already, it can be a problem. Okay, so well, actually we will do that in the genetics, but an overview of alleles and what is an allele. If you take a look here, okay, yes. So you can see here your chromosomes. Your chromosome, normally you have something like this, like two strands of chromosomes, yeah? Basically this is condensed DNA on each other. So if we speak about alleles, for each chromosome, it consists of two alleles, yeah? So these two alleles, you have one allele here, one allele there, and they bind with each other at this point, okay? We'll speak about this point, its importance in the future. But basically, you just need to know that your chromosome, each of your chromosome is made out of two alleles, okay? It's two alleles. Okay. So, yep, it has two alleles, and each gene is located at one allele. So you have gene A is located here. Well, at the other allele, there is also gene A. Okay, so let's take a look at this one. Come on. Why it's not opening? Okay. So, if we speak about the oncogenes, basically becomes actually a tumor that can kill us. But for them to develop this tumor, they need to go under a trigger. A stress must be occurring on the cell to lead to the tumor. This stress can be a virus. It can be some chemical. It can be practically anything. But for the sake of today, we are going to speak about the receptors. So there is something called receptor tyrosine kinase. What is this tyrosine kinase? Basically, it's a receptor located on the cell. And when that receptor is activated, it will activate what? It will activate the secondary messenger. So this is the cell, and it has this receptor tyrosine kinase. It will get activated, secondary messenger gets activated, that will activate the genome. Yeah, this is a double-stranded DNA. And when that is activated, you will have more transcription. Remember, what is a transcription, guys? It's conversion of what to what? Transcription. It's conversion of DNA to RNA. Excellent. DNA to RNA. Translation is the process of conversion of RNA to protein. Yeah? So we have the transcription and the translation. Transcription, conversion of DNA to RNA, and the translation is the conversion of RNA to a protein. So to sum of all of them up, if we say the process of converting a DNA to a protein, which obviously you need to go 2 steps, DNA to RNA and then RNA to a protein. But to make it in short, this process, this whole process, we call it gene expression, okay? So it's a shortcut. Instead of saying, oh yeah, DNA becomes RNA and then RNA becomes a protein, we just say gene expression. Okay, so the secondary messenger stimulates the DNA, which lead to, first of all, transcription, then translation, making many proteins. And also this transcription factor can actually lead to stimulation of the DNA itself to hyperproliferate, which can lead to cancer. So in case of receptor tyrosine kinase, a mutation, the problem is here. Well, it's a gain of function mutation, which means you will have more receptors or less receptors. It's gain of function mutation, more receptors. If you have more receptors, you will have more activation of that cells and more proliferation or less proliferation. More proliferation. That's why the cancer is happening. Well, you are like, okay, more proliferation. What is the big of a deal? Remember when I told you that the more you do something, the more chance you are going to make a mistake? Like, for example, I'm teaching this whole first aid. Of course, I might do one mistake here and there. Why? Because I do it so much. So I have a risk of saying one mistake. Yeah? Between like each 1,000 correct thing I say, there is one mistake. What if I say 10,000 things? There is going to be 10 mistakes. More risk of me saying something wrong. Well, basically it's like someone who's throwing the ball. The more he throws it, the more risk he has to get it one at least wrong. And for cancer to happen, you need just one wrong. See, one allele is wrong and the patient is done. So receptor tyrosine kinase in the lung of the ALK can lead to lung adenocarcinoma. And that's actually important. So lung adenocarcinoma, it can be in a smoker or non-smoker, guys. Yep, we spoke about it yesterday. It can be in a non-smoker and a smoker, actually both of them. But if you have like a patient who has like how do you say it? Like he's a non-smoker and get a lung cancer, you need to think about a mutation of the ALK. By the way, a few of these, I want you to know their names. It's you don't have to memorize them, but if you know what they mean, it becomes easier, you know? So this one, for example, it means the anaplastic large cell lymphoma. You are like, but this is not a lymphoma. Well, there is another lymphoma that is due to tyrosine kinase. So the ALK stands for the anaplastic. What? Somebody write it, breathe. Anaplastic large cell lymphoma. Okay? Lymphoma. Sorry if the dictation is bad. So ALK is anaplastic large cell lymphoma. Okay, so why I'm telling you this? They will not ask you about this, but if you see it, you become more comfortable with it. When you see it, you are like, oh yeah, it's that one. Okay, so the next one is the HER2/neu. The HER2/neu is super important. The HER2/neu is the one that causes breast and gastric cancer, especially the breast cancer. And it's due to tyrosine kinase, receptor tyrosine kinase. And this is even more important. Why? Because this receptor, they invented a monoclonal antibody that can inhibit it. And if you inhibit this pathological receptor, what will happen to the cancer? Will the cancer go away and decrease in size, or it will be enhanced and grow more? If you block that receptor, it will die, exactly. So that drug is actually called trastuzumab. Trastuzumab. So that trastuzumab basically inhibit the what? The receptor tyrosine kinase. And that's why it's important to know it's a receptor tyrosine kinase, because you have an antibody that can function against it. So basically, this is the cell, this is the receptor, which is a receptor tyrosine kinase of the breast. Because in the future, we will speak that tyrosine kinases are different. For example, here they write receptor tyrosine kinase, this one leading to lung cancer, while this one is leading to breast and gastric cancer. So yeah, both of them are tyrosine kinases, but by the shape, they are different a little bit. So comes the question, why they don't make a monoclonal antibody for each of these and we are done with cancer? That sounds logical. Yeah, but there is one problem only. I sent it on Telegram, you can check it out. So, it's really costly. Yep, it's really costly to make a new drug. It costs a lot of money. To make a new drug, any new drug, it costs an average between $3 and $10 billion. Okay? So, it's a lot of money. Well, I will take 1 billion. Okay, so the HER2, a new ERPB. So if we speak about, by the way, there's millions of cancers, like there's so many different subtypes of cancer, so you need to multiply that 3 billions per, like, I don't know, it could be like the whole GDP of the US for the next 10 years. So, from Canby, okay. So, HER2, the monoclonal antibody is called what? It's called trastuzumab. You must know about this. Okay, so the next thing you must know, it's highly related with gastric cancer as well. Okay, so 2 cancers, not 1. And it's a gain of function mutation, not loss of function mutation. Basically, I just want to show you a quick look about how a tyrosine kinase look like. You don't have to know how it looks like, just for the sake of completion. Yes, it has 6 subunits, as you can see here. And when a ligand binds here, it basically leads to what? To overactivation, which lead to downstream signaling pathway, which lead to cell proliferation and migration. I like this picture. Okay, so here you go. So this is when the patient has a receptor tyrosine kinase pathology. Well, they can mention for you something about a non-receptor tyrosine kinase. You are like, what the? Yeah, non-receptor tyrosine kinase. This whole thing, guys, this is a tyrosine kinase. So if you have a mutation that is a non-receptor tyrosine kinase, the pathology is inside. It's in this section, okay? Not outside, it's in the inside. Okay, let's take a look. Good. So receptor tyrosine kinase, that's easy. Breast cancer, gastric cancer. You must memorize all of these. I told you the easy way, just to print it and put it in your bathroom. Every time you are going to the bathroom and you go a lot during the day, you will see it, read one of them. Every single time you go, you will see it and you will know it. How do they ask this? Well, it's actually, they don't ask you exactly from here. It asks you about each system. For example, when I'm going to speak with you about the lung cancer and respiratory, I'm going to mention for you that an oncogene leads to it. So practically, it's memorization, sadly. You need to know that lung adenocarcinoma is related to the ALB, and you need to know that it's a receptor tyrosine kinase causes it. So you need at least 1 allele to be mutated to get it. That's how they ask about it. They tell you how many alleles do you need to be dysfunctional. 1. 1 is enough. While in case of tumor suppressor gene, you need to have both. You will see your worthy question about it. So it's yeah, just print it and put it in your bathroom. Okay, the next one is the rat. So if we speak about the rat is really famous for causing multiple cancers, okay? And it can lead, for example, to men, which is multiple endocrine neoplasia, type 2A and 2B. So 2A and 2B. And also medullary and papillary thyroid cancer and pheochromocytoma, which are actually subtypes of MEN2A and MEN2B. So again, same idea. Receptor tyrosine kinase, you have a receptor, it gets hyperactivated. When it gets hyperactivated, it leads to it leads to its activation. So let me speak a little bit about this. Well, what does it mean, gene products? If we speak about gene products, for example, we have activation of this receptor. This receptor, it's a tyrosine what? Kinase. What does the word kinase mean, guys? Kinase, it means some enzyme or some structure that lead to phosphorylation. Phosphorylation in chemistry means activation. So this phosphate, see, there are 6 of them. It goes and bind to where? It goes to bind to some transcription factor, for example. It can go and bind to the nucleus itself. I don't want to go in details, but there is one that we are going to speak about in the future. If you see this phosphate, it can dissociate and go and activate other substances, okay? So it can lead to further activation inside of the cell. That's what I want to reach. So when that happens, that activation can lead to making of a lot of DNA. So if you had 10 DNA, now you will have 100,000. If you have 100,000 DNA, wouldn't they like to proliferate and make a family and live happily ever after? Of course they would like. So if they like to do this, you will have more proliferation. If you have more proliferation, you will have more nucleic material. And nucleic material is a gene product. Okay, so if you have more nucleic material, what will happen to your nuclear to cytoplasmatic ratio? What will happen to the size of your nucleus compared to your cytoplasm if you have a lot of DNA? Where is DNA stored? DNA is stored inside of your nucleus. So it will be increased ridiculously. That's why if we check any type of cancer, let me be wild, okay, cervical cancer, any type of cancer actually, it doesn't really matter. And if you take a look at this histology, you will be able to clearly see an increased size of the nuclei. Look at the one on the right. This A is normal, D is an actual cancer. If you compare A to D, D has an increased in nuclear to cytoplasmatic ratio, which is leading to the cancer, okay? Good. So the next thing, RET. So RET, I told you it's a receptor tyrosine kinase. They can ask you, well, tell me guys, in case of RET, do I need to have a mutation of 1 allele or 2 alleles? One allele is enough, exactly. Okay, so that's what happens during RET. Okay, the next one is the Pcr Abl. Notice something here. They write non-receptor tyrosine kinase. The first 3 were tyrosine kinases, but this one is different. It's a non-receptor tyrosine kinase. What does that mean? Well, as I showed you already, you don't have always to have the receptor itself. You can have tyrosine kinase, but without a receptor coupled into it, okay? So let's take a look at it. So if you take a look at it, it can be like only the intracellular portion. See, this portion. So you don't have an actual receptor to anything. You can have a growth factor affected, yes, but you don't have an actual receptor to a ligand. Okay, so we call that a non-receptor tyrosine kinase. Okay, so a non-receptor tyrosine kinase basically means it's inside. So if you take a look at this one, it's basically showing you a receptor tyrosine kinase, which is this guy. It has an actual receptor outside, and it's showing you a what? A non-receptor tyrosine kinase, which is inside of the cell. Both of them, when they are activated, they will release phosphate. That phosphate will activate the DNA, which leads to the cell proliferation. Okay, and that can lead to cancer. So the Pcr Abl can lead to a form of cancer that we'll speak about that is really famous, like CML and Allele. And it's super important. Why? Because we have a drug that inhibits it. Anybody know the name of the drug by a chance? Try somebody know the drug that inhibits the cancer in case of CML? No? Okay, so, oh yeah, exactly. It is amitinib. Okay? So amitinib is the one working on it. Anything that ends with tinib, basically. So if you speak about it, it's amitinib, and I just going to send you a picture right now. Okay, so if we speak about yep, we'll send you that picture later. It's exactly how the drug function. It's actually quite interesting. It binds amitinib. Sorry. Amitinib. So the amitinib binds directly to the tyrosine kinase, not the receptor, to the tyrosine kinase itself, which competitively inhibited. And when it inhibits it, when you have a competitive inhibitor at that site, what happens to what will happen to this enzyme? It will become dysfunctional. So it becomes dysfunctional. We have a decrease in the Vmax, which we will study later in pharmacology, and that exerts this drug to become this gene to become dysfunctional. And that's how amitinib work, or anything with a tinib in the end. Okay. I know that this chapter requires like a lot of material outside from first aid, because it's kind of like just every little system in first aid all in one page. That's why I told you to print it. So the next one is about the JAK-STAT. So what is this JAK-STAT? JAK2, it's a non-receptor tyrosine kinase. It can lead to myeloproliferative neoplasms, for example, polycythemia vera, also thrombocythemia, which is an increased number of thrombocytes. So this is related to the JAK-STAT. So if you take a look at JAK-STAT pathway, it goes something like this. So here is the JAK, and when you activate the JAK, as you can see, guys, notice something here. The JAK is not bound to an actual receptor. There is no actual receptor. Yes, it can be affected by growth factor. Okay, so let me put it here. Can you see now? So the JAK is converted to, it activates the STAT. How? JAK is a kinase. It phosphorylates the STAT. When the STAT becomes active, it manipulates the nucleus. When the nucleus is manipulated, it starts making different substances, like the BCL2. What the BCL2 function, guys? Can someone tell me what was the function of the BCL2? Come on. It was related to which organelles, the BCL2? Mitochondria. Yes, it was protecting the walls of the mitochondria. And if you protect them too much, you will get more survival, more proliferation, more risk of cancer. It also can lead to down regulation. As you can see, this one is in red. Down regulation, yes, for apoptosis. So when you have more BCL2, you will have less apoptosis, more risk of proliferation. Okay, if you have less P53, that's actually a tumor suppressor gene that we'll speak about later. But when you have it less functional, the cell cycle will be going faster. Okay, and the next one is this one, the BRAF. The BRAF, sadly, I don't know why they did this, but you must memorize it as serine threonine kinases, which actually lead to direct activation of the DNA, which can precipitate the patient to getting different cancers like melanoma, non-Hodgkin's lymphoma, colorectal cancer, papillary thyroid cancer, and hairy cell leukemia. Sadly, they are not related to each other, these cancers, but you must memorize them. That's why I repeat again, print this freaking page and put it somewhere. So the c-KIT. So the c-KIT. So it's cytokine receptor, and it's in the gastrointestinal tumor, stromal tumor, and a disease called mastocytosis. What is mastocytosis? First of all, I would like to tell you about the symptoms of mastocytosis. It's a patient that will come to you that have symptoms of a lot of like histamine in his body. So they will have rash, they will have diarrhea, and they will have ridiculous urticaria, as you can see. Yeah, they will also have really bad abdominal pain as well. When you do a skin biopsy, you will see a lot of mastocytes. And when you see these mastocytes, done, you have confirmed it. And it's basically a cancer, and actually it's a really bad one. Okay? So, yeah. Basically, they have diarrhea, urticaria, and you diagnose it with a biopsy of the skin that will show you what? It will show you mastocyte. Okay. Okay, so let's speak about we will speak about mastocytosis, GI. I think GI, we'll speak about it in GI. Because you have to differentiate between it and carcinoids. They have different symptoms. Okay, c-MYC. So what are c-MYC, MYC? All of these MYCs are a transcription factor. And according to the first letter, you can determine which one it is. For example, the C. I just remind myself so that like the C in English, it's mostly pronounced as a K, yeah, like a cat, for example. Percat lymphoma. So it's cat with a C. So I imagine it cat MYC. So the cat MYC is the Bercat lymphoma. Okay? It's a transcription factor. Remember the Bercat lymphoma, it was a patient coming from where? In which country Bercat lymphoma is Bercat lymphoma mostly comes from Africa, mostly in the jaw of the patient. And yeah, while the next one is the MYC-L or L-MYC, L for lung cancer. And the MYC-N is for neuroblastoma. There is one symptoms of neuroblastoma they keep putting on the exam, and it's not in first aid, and you will not be able to see it anywhere except on the real exam day. I think it was mentioned in New Word, but between the lines, and this is really annoying. So normally, when you see the raccoon eyes, what does it mean, the raccoon eyes, guys? The raccoon eyes mostly are seen in patient with not big eyes. You can see like it's black under the eyes, around the eyes. And it's due to basal lung fracture, okay? Basal cranium fracture. That's what raccoon eyes mean. But raccoon eyes also can happen as a paraneoplastic syndrome, which I will explain later what it is, in some patients who have, you guessed it, neuroblastoma. So neuroblastoma can be seen in some cases. Neuroblastoma can show raccoon eyes in some patients. So if you have a patient with abdominal mass and no trauma, having raccoon eyes, you think about neuroblastoma. Okay. Next. So the next one is the KRAS. So the KRAS, I told you already about this. I told you that the cell has a receptor, and that receptor can be activated by a growth factor or whatever, any ligand. And then it has a secondary messenger, which was RAS. Here comes RAS. So if this RAS is hyperactivated, like in case of KRAS, the RAS GTBase will be activated, and that will lead to hyperproliferation of the cell, which can lead to colorectal, lung, and pancreatic cancer. Well, guys, is pancreatic cancer serious? Yeah. How long do you think a patient have after you discover pancreatic cancer in him? Yeah, 6 months, and some of them like can live up to a year if you do something called the Whipple procedure. A Whipple procedure, basically it's crazy. So there is something called the Whipple procedure that can prolong the life of the patient. So a question comes like this. So this Whipple procedure, basically what do you do for the patient? Well, you cut half of his stomach, you cut his his gallbladder. He does not need that. Remove the duodenum, remove a portion from the intestine, and then connect everything together. So as you can see, remove the stomach, gallbladder, common bile duct, half of the pancreas, all of the duodenum, some of the jejunum, and you do it a Frankenstein style and connect everything together. So a question comes, yes, it prolonged the life for 6 months, but is it worth it? Because, you know, for recovery from such surgery, it's really a long period of time of recovery. So he will be staying in a bed for 6 months. He will not be able to do anything. It's not worth it except in one case, a pregnant lady. If you have a pregnant lady, a case of a pregnancy, a Whipple procedure should be kept on the table. Why? Because you want to allow her to deliver that child before she passes away. So in that case, you would propose a Whipple procedure. In a pregnant lady, before she, you tell her, okay, look, you can prolong your life to get that child if you want. So the last one is the BCL. BCL, you remember when we spoke about apoptosis, I told you about the pro-apoptotic proteins, molecules, and an anti-apoptotic molecules. Can anybody tell me a pro-apoptotic molecule? Any pro-apoptotic molecule? The BAC and the BAX, which used to activate the caspases. Excellent. So the pro-apoptotic molecule used to be the BAC and the BAX. And the anti-apoptotic molecules used to be the BCL and the BXL. Let us take a quick look at them. So here you have the pro-apoptotic molecules. Yeah, here they are. And here you have the anti-apoptotic molecules. So again, BAC and BAX are pro-apoptotic. And BCL and BCL2 and BCLXL are anti-apoptotic molecules. And the function of this BCL actually is just to prevent the BAX and the BAC from killing your mitochondria. What happens there is actually quite interesting. If this is your mitochondria, it has the cytochrome C inside of it. And there is a particle that is protecting your mitochondrial wall, okay, which is BCL2. The BAC and the BAX always wanna kill it, but BCL is preventing them from doing so. She's like, not when I'm here. So if you have overexpression of the BCL2, your cytochrome C will be always protected. So apoptosis cannot occur. If you do not have apoptosis, you will have hyperproliferation of the cell, which is practically cancer. So, just for fun, cytochrome C is located in the inner mitochondrial membrane. You will be able to see it when we discuss biochem together. Come on, biochem. It's in the metabolism chapter. You can take a look at it exactly right here. You can see the cytochrome C. This is the guy that is getting released, which activates the caspases and lead to apoptosis. Okay. Okay, so you can see the BCL, it's an anti-apoptotic molecule that inhibit the apoptosis. So it can precipitate to follicular and diffuse B-cell lymphomas. So an overproduction of the BCL2 is not a good idea. That's what I want to reach for you. Okay, really a quick recap. Oncogenes, do you need to have a mutation of one allele or two alleles? One. Is it a gain of function mutation or loss of function mutation? Exactly, it's a gain of function mutation. Okay, so we said it can be a problem in the tyrosine kinase, non-receptor tyrosine kinase, inner signals, and the transcription factors. Also, the RAS, which is a secondary messenger, or a defect in the apoptosis. For now, I'm going to give you guys a break because this is a lot to handle. I know this topic is one of the hard ones. And next, we are going to speak about the tumor suppressor gene. Today, we'll be done with the whole neoplasia chapter and speak a little bit about renal. Yes, I can speak really quick now just about the gain of function mutation one more time, and it goes something like this. So if we have a cell and it has this receptor, if you activate this receptor, it becomes it hyperproliferate, yes? And this hyperproliferation is the thing that is leading to cancer. So to get cancer, you need to have a gain of function mutation, which means the receptor becoming more active, or loss of function mutation, which means the receptor become inactive. Yes, all proto-oncogene is a gain of function mutation in sense of the proto-oncogene is converted to oncogenes. Oncogenes can make you, for example, more receptors to lead to the cancer, more secondary messenger to lead to the cancer, more transcription factor to lead to the cancer, more kinases to activate other enzymes that lead to the cancer, or more RAS GT bases to lead to the cancer. Or again, function mutation can convert the proto-oncogene to oncogene to make you more BCL2, which will overprotect your cell from apoptosis and can lead to the cancer. So a gain of function mutation is a form of a mutation that converts a latent proto-oncogene to an actual oncogene that can lead to cancer by different mechanisms. Okay, make sense? Okay, so if it does not make sense, let me know after the break and I will re-explain it. So for now, just try to freshen up. Let's meet at 7 and yeah, see you. Remind me to start the recording after the break, please.